Usually when we talk about a medical diagnosis, there's this expectation of structural precision. It feels like engineering.
Right. Very mechanical.
Yeah. Like you break your arm, the x-ray shows that jagged white line, and the doctor just points at the screen and says, there it is. That's your problem.
It's comforting in a way. I mean, the binary nature of it. It's either broken or it's not.
Exactly. But then you step into the world of immunology, especially when dealing with rare genetic diseases, and, well, suddenly that trusty x-ray machine is just totally useless. We find ourselves looking at a diagnostic landscape that is, honestly, incredibly murky.
Oh, it is the absolute definition of diagnostic muddy waters. You're no longer looking for a broken bone. You're looking for a broken whisper between two microscopic cells in a sea of billions.
Which is exactly why we are here today.
Uh-huh.
Welcome back to the Deep Dive. If you're listening to this, maybe you're prepping for clinical rounds or trying to catch up on the latest shifts in immunology, or maybe you're just insanely curious about the hidden mechanisms of the human body.
And we have a fantastic one for you today.
We really do. Today, our mission is to completely unpack a dense, incredibly fascinating 2026 research article from the journal Immunity. It's titled, NK Cell Dysfunction and Interferon Gamma Production Underlie Autoinflammation in Mevalonate Kinase Deficiency.
Quite a mouthful, I know.
Right. But we are going from top to bottom, translating all that high-level jargon into the actual story of what is happening inside the cell. We're going to explain every single detail.
It really is a remarkable paper because it solves a profound biological mystery involving broken cellular conveyor belts, frustrated microscopic assassins, and ultimately a groundbreaking treatment pivot that actually saved a critically ill infant.
Yeah, that case study is wild. Let's set up the core mystery we are exploring today for everyone listening. How does a tiny glitch in the way our cells process fats, literally a microscopic lipid assembly line defect, lead to a massive, life-threatening, hyperinflammatory cytokine storm in the entire body?
It sounds totally counterintuitive.
It does. To understand this disease, which is called mevalonate kinase deficiency, or MKD, we actually have to start on the factory floor of the cell.
Right. So we have to look at a foundational piece of cellular machinery called the mevalonate pathway. This is a ubiquitous metabolic pathway inside all our cells.
And ubiquitous meaning it's everywhere.
Exactly, yeah. Its primary job is to synthesize these very specific long-chain fats called isoprenoid lipids. Specifically, we're talking about raw lipid materials like farnesyl diphosphate and geranyl diphosphate.
Okay, those are some serious chemistry words.
They really are. But the reason the cell spends so much energy in making these complex fats is because they are crucial for a process called protein prenylation.
Okay, let's unpack that, because protein prenylation sounds incredibly dense. I like to imagine the inside of the cell as like a massive, bustling shipping warehouse.
I love this analogy.
You got all these worker proteins, right? Specifically these small ones called Rab and Rho GT passes. And their entire job is to move cargo around the cell.
But to do their job, they need to be able to attach themselves to the lipid membranes of the cellular compartments.
Right. So prenylation is basically the process of slapping a sticky barcode and a physical lipid tether onto the package. Without that lipid anchor, the worker protein can't grab onto the membrane. It just sits on the fluid floor of the cytosol, totally useless.
That visual hits the nail perfectly on the head. And we aren't just talking about one or two workers here. Over 300 different proteins in the human body rely on that specific sticky barcode to function.
Wow. Over 300.
Yeah. Now, in mevalonate kinase deficiency, the disease we're looking at, patients are born with biallelic loss-of-function mutations in the MVK gene.
Meaning both copies of the genetic blueprint for that specific factory enzyme are completely ruined.
Exactly. Because both blueprints are defective, the mevalonate kinase enzyme doesn't work. The assembly line jams up very early in the process. Those essential lipids, those tethers, are simply never made.
So going back to the warehouse, the floor just starts filling up with these unprenulated Rab-GT pases. They are floating around in the cellular fluid, completely unable to do their shipping jobs.
And the fallout from that is severe. Clinically, this disease presents on a spectrum, which is graded by the amount of residual enzyme activity the patient has left.
So it's not always identical in every patient?
No, not at all. The milder form is called HIDS, or hyper-IgD syndrome. These patients might have somewhere between, say, 1 and 20% of normal enzyme activity.
Okay, so they have a little bit of the factory working.
Right. And they typically suffer from episodic fevers, skin rashes, joint pain, and swollen lymph nodes. But the severe form, known as mevalonic aciduria, is absolutely devastating.
That's the one with less than 1% enzyme activity, right?
Yes, less than 1%. They face severe developmental issues, facial dysmorphia, and life-threatening systemic inflammation.
Wait, let me challenge the mechanics of this for a second, because I think you listening might be wondering this too. If the cells are structurally failing to build these lipid anchors, and their internal shipping logistics are ruined, why doesn't the cell just initiate programmed cell death?
It's a great question.
I mean, if a factory's broken, you shut it down. Why does this broken metabolic pathway result in massive whole-body inflammation?
That was the exact paradox stumping researchers for decades. The historic assumption in the medical community was that macrophages, the big eater cells of the immune system, were just churning out a pro-inflammatory alarm chemical called interleukin-1 beta.
So they thought the macrophages were just panicking because of the cellular stress.
Exactly. But that theory had a massive hole in it.
Because the treatments didn't work.
Right. When doctors gave these patients standard drugs that block interleukin-1 beta, drugs like anakinra, many MKD patients completely failed to respond.
The inflammation just persisted.
It persisted, destroying their tissues. This told researchers that interleukin-1 beta wasn't the main driver. We were missing a much bigger, much more dangerous piece of the puzzle.
Okay, so let's do a quick 5-minute recap and bullet summary for the audience to lock down this first segment.
Yeah.
Bullet 1, MKD is caused by a broken mevalonate pathway.
Yep. Bullet 2. Proteins, specifically Rab GTpases, lack their lipid anchors. The prenylation fails.
And bullet 3. This leads to a buildup of useless proteins and severe whole-body inflammation that we use to completely misunderstand.
That's the perfect summary. It forces a total perspective shift, which is why the researchers of this 2026 paper decided to start from scratch. They engineered mutant mice to carry the exact same MBK gene mutations found in human patients.
So they built a living model of the disease.
Exactly. And instead of just guessing which cells were causing the inflammation, they used single-cell RNA sequencing.
Oh, I love this tech. For those listening who might not be in the lab every day, single-cell RNA sequencing is basically like taking a high-resolution census of every single cell in a tissue.
Yeah, it's incredibly precise.
It tells you exactly what genes each individual cell is turning on or off.
It provides an unprecedented level of detail. And when they looked at the immune cells in these mutant mice, the result was completely unexpected. They found a 60% decrease in the frequency of natural killer cells.
Wait, natural killer or NK cells? These are the body's primary viral assassins.
Yes.
And they were just, what, missing?
A massive portion of them, yes, gone. But to understand why, the researchers had to look at the life cycle of these cells. They didn't just count them, they looked at specific protein tags on the outside of the cells.
Think of them as biological ID badges that tell us how old or mature the cell is.
Exactly. Normally, as an NK cell matures, it swaps out a trainee ID badge for a veteran ID badge. In the paper, they tracked this very specifically. In mice, they looked for the loss of a marker called CD27 and the gain of one called CD11B.
Okay, so CD27 is the trainee badge, and CD11B is the veteran badge for the mice.
Right. And for humans, the mature veteran cells have the CD56-DIM, CD57 markers. But the concept is exactly the same across species. The cells need to graduate.
But in the MKD mice, the census showed almost none of the cells had the veteran badge. There was a huge excess of immature cells just stuck with the trainee badge.
Yes. They then looked at blood samples from human MKD patients and confirmed the exact same phenomenon. The mature NK cells were drastically reduced. They were arresting in development.
Here's where the biology gets really intense. Why are they failing to graduate? NK cells have to go through a massive proliferative burst to mature. I always think of it like an intense military boot camp.
That's a good way to put it. It requires immense cellular energy to build up their toxic arsenals.
Right. To figure out why they were dying during this boot camp, the researchers used a special dye called mito-SOX.
Yes, mito-SOX. It essentially lights up when there is high mitochondrial oxidative stress in the cell.
And what did they see?
The mutant NK cells were practically glowing. They were completely overwhelmed with oxidative stress.
Let's connect that back to our warehouse analogy for a second. Normal cells constantly clear out damaged mitochondria, at their little power plants, through a biological trash removal process called mitophagy.
Right, taking out the cellular trash.
But to take the trash out, you need those shipping logistics to be working.
You need the Rab GTpases,
Yeah.
which, as we know, are sitting uselessly on the floor because the mevalonate pathway isn't giving them their lipid anchors.
Oh, wow. So the whole system cascades.
Exactly. Without prenylation, the trash removal system completely fails. The damaged mitochondria pile up, toxic cellular exhaust floods the cell, and these vital immune assassins basically die of toxic stress before they ever graduate boot camp.
Okay, let's do our next quick bullet recap here. Bullet 1. Single-cell RNA sequencing revealed a massive drop in mature NK cells in both mice and humans.
Bullet 2. Specific cellular markers, like CD57 in humans, prove these cells arrest before maturing.
And bullet 3. High mitochondrial oxidative stress, shown by that mito-SOX dye, is likely killing them off. So if you're listening to this, think about the implications. If your body's specialized frontline defenders are dying off before they hit the battlefield, what happens when something as simple as a common cold virus enters the body?
The host is left incredibly vulnerable. But here is the really tricky catch. It's a 60% decrease. That means some NK cells do survive the stress.
Right, 40% make it through.
Yeah, some of them manage to mature and circulate in the blood.
And that leads to the next huge question. If these few cells actually survive the boot camp, how are they supposed to fight? I mean, they still have broken shipping logistics. Don't they need those RAB shipping proteins to deliver their weapons?
That is the perfect question, and it's exactly what the researchers wanted to know. To answer it, they used live cell confocal microscopy to watch these surviving NK cells interact with infected target cells in real time.
I love this part of the paper. This is what we call the jammed gun mechanism. Let's break down how a healthy NK cell normally takes out a target.
In a healthy scenario, an NK cell recognizes an infected cell and binds to it, creating a physical point of contact. We call that the immunological synapse.
It's like a docking port.
Exactly. Inside the NK cell, a logistics hub forms called the microtubule organizing center, or MTOC. The NK cell then uses its RAB shipping proteins to transport toxic payload packages. These are cytolytic granules filled with destructive enzymes like granzyme B along the microtubule tracks right to the synapse.
And then it just dumps the toxins into the target cell, destroying it completely.
Yep, clean kill.
Okay, let's build on our visual from earlier. Imagine a highly trained sniper. They spot the target, they lock on. That locking on is the MTOC forming and aiming at the synapse, but the mechanism to actually load the bullet into the chamber is jammed. The internal gears, those RAB proteins, are missing their lipid grease. So when researchers looked at the live imaging of the MKD cells, they saw the MTOC form. The sniper was locked onto the target perfectly.
But the toxic granules were just scattered all over the back of the cell. They couldn't move them to the synapse.
The cell is structurally impotent.
Yes. When they ran cytotoxicity tests, basically measuring how well these immune cells could actually kill lab-grown target cells.
And they used specific targets for this, right?
They did. They used YAC1 cells for the mice and K562 cells for the human tests. And across the board, they found a 30 to 40% reduction in actual physical killing power.
So the MKD NK cells can recognize the threat, they can bind to it, but they physically cannot deliver the lethal blow.
They are jammed.
Time for another quick bullet summary. Bullet 1. NK cells use RAB proteins to transport toxic granules to target cells.
Bullet 2. Live imaging shows MKD NK cells bind to targets but fail to deliver those granules.
And bullet 3. This results in a 30 to 40% reduction in physical killing power. So put yourself in the shoes of that specific immune cell for a second. You are circulating in the blood. You recognize a deadly virus. You grab onto it. You lock your sights, but your primary weapon physically will not fire. How does a cell handle that? How does an immune cell call for backup when its primary weapon fails?
Well, it panics. And that panic translates into a massive biochemical reaction. Because the NK cell is locked onto a target it cannot kill, the situation quickly devolves from a targeted, quiet assassination into a loud, systemic alarm.
So the cell compensates for its physical weakness by screaming for help chemically.
Yes. The researchers measured the biochemical fallout of this prolonged, frustrated engagement. When these MKD NK cells bind to a target, they show abnormal spilling of their granule.
Like they're leaking.
Basically, yeah. They start leaking out the sides, which the researchers tracked using a surface marker called CD107A. But far more dangerously, these frustrated cells begin heavily secreting inflammatory cytokines.
The alarm bells.
The loudest alarm bells. Most notably, they flood the surrounding area with interferon gamma, along with tumor necrosis factor alpha, and GM-CSF.
So they can't stab the virus, so they set off the biological fire alarm. They just hold the alarm button down.
And the researchers proved this was the specific trigger for the disease flares by running a viral challenge. They introduced a pathogen called mouse cytomegalovirus, or MCMV, to the mutant mice.
And what happened? I'm guessing it wasn't pretty.
It wasn't. The defective NK cells couldn't clear the virus. Because they couldn't clear it, they stayed locked on, getting more and more frustrated, and they just kept secreting interferon gamma.
Just a continuous loop of panic.
Exactly. The mice developed astronomical levels of interferon gamma in their blood and suffered severe organ-damaging inflammatory pathology. They were dying from the inflammation, not just the virus.
And it gets even more profound because it's not just live viruses that cause this. The researchers managed to trigger this exact same cytokine storm using common vaccine adjuvants.
That's a very crucial detail. Adjuvants are substances like CpG or IFN-γ, which are used in routine human vaccines specifically to wake up the immune system and say, hey, look at this.
Which perfectly and tragically explains a known clinical reality. Human patients with MKD often suffer massive, life-threatening inflammatory flares right after routine childhood vaccinations or incredibly mild respiratory bugs.
Because their broken NK cells see the harmless vaccine adjuvant, they try to engage, their shipping mechanisms fail, and they just flood the child's body with interferon gamma.
It is a localized, microscopic cellular frustration that spirals outward into a systemic hyperinflammatory state. Let's recap this segment. Bullet 1. Unable to kill targets, NK cells hypersecrete interferon gamma and other cytokines.
Bullet 2. Mice infected with the virus show massive inflammation and a total failure to clear the virus.
Then bullet 3. This mechanism finally explains why real-world MKD patients suffer massive flares after minor infections or vaccines. If you are listening to this, does it change how you view inflammation? We often think of inflammation as an overactive, overly aggressive immune system. But here, it's actually the body's backup alarm system going haywire precisely because the primary defense is too physically broken to finish the job.
It's a total paradigm shift in understanding autoinflammation. And more importantly, it fundamentally changes how we treat the patients who are actively dying from it.
Which brings us to the most powerful part of this paper, the clinical application. The researchers didn't just leave this as a fascinating lab discovery. They took this entire molecular journey from lipid assembly lines to single-cell sequencing to live imaging of frustrated snipers, and they brought it directly to the bedside.
They applied it to a harrowing clinical case. The patient was a male infant, born premature at just 30 weeks and 3 days, weighing barely 1,600 grams.
That is so small.
Incredibly small. And even in the womb, ultrasound showed he had fetal hepatosplenomegaly, an enlarged liver and spleen. Right after birth, he became critically ill with fever and severe cardiorespiratory failure.
The diagnostic team had to move incredibly fast. First-line tests showed a hyperinflammatory response with signs of a dangerous condition called HLH, which is basically an immune system gone rogue, attacking the body's own organs.
Right. They ran urine metabolic testing, which flagged suspected mevalonic aciduria. Then, rapid exome sequencing confirmed the infant had those biallelic MVK mutations we've been talking about.
They even ran specialized lab tests on the infant's blood, which confirmed a massive buildup of unprenylation Rab proteins. The lipid shipping labels were definitely missing.
So he clearly had the severe form of MKD. The doctors immediately tried the standard treatments. They hit him with high-dose steroids. They gave him high-dose anakinra to block interleukin-1 beta.
Which, if you remember from the beginning of our dive, was the old theory of what caused the inflammation.
Right. But it wasn't working. The infant was failing, his gut was heavily inflamed, he was constantly irritable, his markers weren't dropping, and the inflammation was just relentless.
But because this research team now understood the true mechanism, they knew that interleukin-1 beta wasn't the primary driver, and that interferon gamma, the scream of the frustrated NK cell, was the real culprit. They made a massive pivot. They administered a drug called baricitinib.
Let's explain why that matters. Baricitinib is part of a class of drugs called JAK inhibitors. What it does is block the downstream signaling of interferon gamma.
It essentially cuts the wires to the fire alarms so the sirens stop ringing.
Exactly. And the clinical results were astounding. The infant's inflammatory markers, specifically his CRP and ferritin levels, absolutely plummeted.
Wow.
Yeah. His severe clinical symptoms, the unrelenting fever, the severe gut inflammation they resolved, he stabilized.
Wait, I have to push back here because immunology is always a double-edged sword. If we block interferon gamma with a JAK inhibitor, aren't we artificially suppressing the infant's immune system even further?
That's the obvious concern, yeah.
I mean, if his NK cell already can't fight a virus because their guns are jammed, and we step in and mute their call for backup, how does the patient survive the original infection?
That right there is the delicate, terrifying tightrope of clinical immunology. Yes, there is absolutely an immunosuppressive risk when you use a JAK inhibitor, but you have to look at what is actively killing the patient in severe autoinflammatory conditions like MKD.
The inflammation.
Exactly. The infant wasn't dying from a massive viral load. He was dying from the alarm, the cytokine storm. The inflammation itself was literally destroying his internal organs.
You have to silence the alarm to save the house from vibrating itself to pieces.
That's a great way to put it. By using buricitinib to suppress the interferon-γ pathway, they stopped the lethal hyperinflammation, which bought the child time to stabilize and heal.
So last bullet recap here, bullet 1. A severe infant case of MKD presented with life-threatening inflammation.
Bullet 2. Standard interleukin-1 beta blockers, like anakinra, failed to fully control the disease.
And bullet 3. Based on the new interferon-γ discovery, doctors used buricitinib to successfully suppress the inflammation and save the child, translating a lab discovery all the way to a saved life. Think about the sheer power of single-cell sequencing in that context.
It really opens up a whole new frontier for personalized medicine. And if we step back and look at the broader picture, MKD isn't an isolated anomaly. It is just one disease in an emerging broader family of disorders called prenylopathies.
Meaning diseases caused by a failure to prenylate or attach lipids to proteins.
Right. There are other genetic defects like PMVK deficiency that affect different specific enzymes in that exact same mevalonate lipid pathway. What we learned here today about NK cell frustration, defective granular trafficking, and interferon-γ hypersecretion will likely rewrite the medical textbooks and treatment protocols for all of these related diseases.
It completely changes how we view the system. We used to think of the immune system as this simple, blunt force army that just attacks things. But this deep dive shows us that it is actually a highly interconnected, incredibly sensitive sensory network.
It really is.
When one small, purely structural mechanism like a lipid shipping label on a microscopic protein gear fails, the entire network misinterprets the failure, panics, and goes into overdrive.
It perfectly highlights how local microscopic failure leads to macroscopic systemic chaos.
Which brings me to a final provocative thought for you, the listener. We just spent this entire time unpacking how a tiny structural glitch in one specific cell type causes a massive, previously unexplained fever and cytokine storm. So looking beyond rare genetic diseases, think about the general population.
Okay, where are you going with this?
Think about conditions like chronic fatigue syndrome, mystery autoimmune flares, or severe unexplained reactions to routine viral infections.
Oh, conditions that we often treat with broad immunosuppressants because we don't know what else to do.
Exactly. What if some of those conditions are actually driven by microscopic, local cellular frustrations? What if an immune cell somewhere in the body is trying to do its job, physically failing, and quietly holding down the fire alarm? Are we looking at the wrong cells when we try to treat these everyday mystery illnesses?
That is a fascinating thought. If the underlying gears are jammed, we might be spending all our time trying to put out a fire that is constantly being reignited by the body's own broken, frustrated alarms.
The diagnostic X-ray machine for the immune system might still be murky, but studies like this finally let us turn on a flashlight and see the exact jammed gear in the dark. Thank you for taking this incredibly dense, rewarding journey into the cell with us today. Keep questioning the mechanisms, look for the frustrated cells, and we'll see you on the next deep dive.